your table, it looks like this. You are going to be working in groups of 2 today. We are going to be solving a crime. There was a crime in Bronzeville last night. Actually, I don't know if it's in Bronzeville. I'm saying it's Bronzeville because we're in Bronzeville. What I need right now, or what I would like, I need someone who has the best CSI voice to please read the intro. The best CSI voice. Who has the best? We're going to try with Mr. McRae. But the best voice, best SPA voice. We're going to try it. If I have your attention back, please. 5, 4, 3, 2, 1, and 0. Mr. McRae is ready. He's already popped his collar, his detector collar. Can you please read? 69%. It's too loud. I'm going to wait.
It has your Sean read, man.
Thank you so much. For all the right respect, Mr. McRae, as he reads the intro to the slab, there shouldn't be any talking at this time. This is level 0. You may begin, Mr. McRae.
All right, guys, today, the crime report.
Hold on, wait, before you start.
Mr. Scott, can you let him finish, please?
No, wait.
No, can you?
Right before you start.
No, can you please let him finish? I'll take your question in a second. Thank you. I just said it's
It's
level 0.
related to this stuff.
Mr. Scott, please be patient and wait until Mr. McRae is back. I'm sorry, Mr. McRae, please.
All right, guys. The crime report for today. A small pool of blood and a weapon was found near a garbage dump. After examining the area, the CSI on the scene discovered a body in the garbage. And identified him as Ernest, nicknamed One Eye Earl. Earl had a wound to his chest that will be analyzed by the medical examiner. The CSI tested blood samples from the blood pool and the weapon at the crime scene. It was determined that it was human blood, but he needs to know the type of blood to help identify if it was from a victim or the person who murdered him. He has come up with 3 suspects that either knew the victim or were seen in the area before the body was discovered. He would like to question them further while he waits for the DNA test results.
Excellent. Let's give it up for Detective. So, what we are going to be doing today, if you recall from our blood typing lab in the review at Mr. Howard last week, we were taking samples of blood, fake samples of blood, putting them into antibody serum test tubes, and determining what the blood type was. We are going to be doing the same thing today, except we're going to be doing it live. And we're going to do it to solve the crime. But first, I want to make sure that we go over all the blood typing one more time because we haven't done it in a few weeks. So, yeah, actually, I'm going to have a ton of volunteers. The first thing I need is 2 volunteers who can help me at the board. Mr. Nichols, Mr. Jefferson, thank you so much.
Of course you'll pick them.
Sorry, Mr. Miller, you want to volunteer for the first answer?
He's sad.
No, not yet. So, if I get everyone's attention to the board, please, in 5, 4, 3, 2, 1, and 0. Thank you. You may notice that there is a data table that we're about to fill out. It's also on the top right-hand corner of your sheet. So we will be filling this out together as a group. So just a review from last week. Type A red blood cells have only type A proteins on the outside. B red blood cells have B proteins on the outside. AB red blood cells have both A and B proteins. And O red blood cells have no proteins. When we see agglutination or clotting, when we add random blood samples into these test tubes, that is how we know that an antibody has found its matching protein. Mr. Miller, there shouldn't be any eating in class. This is one. Thank you. Please put it away. Mr. Miller. Thank you so much. When we see clotting in each of these test tubes, that means that the antibody has found its matching protein and it sinks all the way down to the bottom of the test tube. So what I would like Mr. Nichols to do, could you please show clotting in the anti-A test tube and no clotting in the anti-B test tube? Mr. Jefferson, can you help me figure out what kind of blood type this is?
Okay, so when an antibody latches onto a blood cell, then would it be positive or negative? The anti-A would it be
By
positive or negative?
Someone with a hand.
Blood type B.
It
No,
would
I
be,
asked him to lead the discussion. Thank
it
you,
would,
Mr. Ferguson.
it would be positive.
I just made you laugh.
It would be positive.
What would be positive, A or B?
A. Ain't that what's
Excellent. Yep, that's positive. Nice job, Mr. McRae.
Alright, so one
Okay.
blood type B, right?
And when an antibody isn't attached to any proteins, which is shown as antibody B, would it be positive or negative?
Negative.
Wow.
Okay, so someone with a hand, by this being positive and this one being negative, can you tell me what blood type it would be?
Based on the chart.
Someone with a hand want to volunteer a potential blood type?
Mr. Junior,